Hey, good evening, everyone, and thank you for joining us for tonight's State of Play. I'd first like to acknowledge the traditional owners of the lands upon which we meet tonight and to pay our respects to elders past, present, and emerging. So for tonight's webinar, we have two exciting speakers lined up. They are Sophia Luikinga and Azin Amin. So Sophia completed her PhD in 2019 at the Florey Institute, and last year she moved to Queensland to pursue her work in the involvement of fats in M&D, and she has been awarded a Scott Sullivan Research Fellowship supported by the M&D and Me Foundation and M&D Research Australia to tackle this over the next 3 years. Azin, on the other hand, completed her PhD in 2022, so a little bit younger than Sophia. She did this at the University of Melbourne. Last year, she was awarded the prestigious Bill Goal M&D Research Fellowship from M&D Research Australia to advance her research on developing an innovative peptide-based therapy for M&D. So without much further ado, I will introduce Sophia to present on her fellowship. And once Sophia is finished, we'll hear from Azin. Sophia?
Thank you so much for that kind introduction, Shu. I will share my screen. Yeah, I think I've got the right one shared. That the right? Sorry, is that the right one?
No, it's the wrong one. You've got to.
I just practice this.
Yeah. So if you shared the slideshow. This is,
I'm so sorry. Yes.
there we go.
Sorry for that.
Thank you, Sophia.
It went right the first time, and then the second time, obviously, it didn't. So yeah, first of all, Shu, thank you for introducing me, and thanks for everyone attending tonight. And I also like to thank M&D Australia and M&D Foundation for their kind support in my work. So I'll be talking tonight about if fats in the blood of people living with M&D, if they are important in the clinic. So why do we need fats? So fats help brain development and functioning, and almost 60%, so the majority of the brain is actually made out of fat. They are structural components of the cell. So for example, if we imagine if you have seen a neuron or a motor neuron, if you see them, and kind of the membrane around it is all made out of fat. So if something changes, it could change the whole functioning of the cell. We also need them to support the absorption of vitamins, and we mainly also use them as energy. So they're very important. And why would we study them in M&D? So we see a lot of fat loss in people living with M&D. And we also know that less fat is linked to shorter survival. So I'll show you a little diagram and only focus on the blue part of this man. So when we look at fats, we see decreased body weight with worsening disease. Decreased BMI correlates with poor prognosis, and a loss of fat-free mass correlates with shorter survival. On the other hand, greater fat mass is associated with longer survival, and we also see increased fat content in the muscle as disease progresses. So there's a lot of things that fats are involved in body-wide in M&D, not just in the brain or spinal cord. So when we have a look at fats, so I just made a few little fat molecules here. They have long tails, they have short tails, they have really complex makeups. And if this fat is completely different from this fat, but on the eye, you can't really tell, but functionality, they are very different. So because of this complexity, for a long time, people have put it in the too hard basket. But over the last decade, there's been new ways to study fats. So this is a little bit of a flow chart, how we have been studying fats. So we can take samples from humans, or I've also been working a lot with preclinical mouse models. And we take these, we can take tissue samples, we can take blood samples, and we extract the fat, and we put them through this really fancy machinery that can spit out all this data for us, and then we can analyze how fats are different from controls, and which fats are different, and also within which fat, how different the composition of the fat is. So we can use it in different ways, these results, as a diagnostic or prognostic tool, but also we can use this dysregulation as a disease mechanism research, and discover maybe new targets for drugs. So for the purpose of today's talk, I'll be talking about the diagnostic and prognostic tool side of things, and I'll talk to you a little bit more about that, because we're going to talk a little bit about biomarkers. And for MND in biomarkers, there's multiple biomarkers. There's not one biomarker for MND that can shorten the diagnosis, that can predict the prognosis, or can tell us if a drug is effective or not. So to date, just summarize some of the biomarkers that are either in research or are available. So we have early on in MND, we see hyperexcitability that we can measure with transcranial magnetic stimulation. We can take cerebrospinal fluid, but that's very invasive, and no one really likes to get a lumbar puncture all the time to look at all these things. But we can look at neurofilament, and I'll tell you a little bit more about that later. We can look at genes and proteins, and also fats. But what most people will know is that with the diagnosis of MND, most people would have had a nerve conduction study and an electromyograph. So a combination of most of these biomarkers is usually used to diagnose MND. We can also take tissue biopsies. So some people in our lab can take biopsies of the muscle, and then they can grow M&D muscles, and they look a lot different than control muscles. And it's really cool techniques that people have been doing. And they've also, in Shu's lab, they've been looking at actigraphy, so just your smartwatch, and looking at little movement changes, if that can predict disease prognosis. So there's some really cool things in the pipeline in regarding to biomarkers. But I want to focus on blood biomarkers, because that is usually quite easy to do. And what people usually think of when they go to the GP, oh, I'm just going to get a blood test done and get it back, and I know what's going on. And so this is a depiction of neurofilament. So when a neuron or a motor neuron is damaged, it releases neurofilaments in the CSF, so cerebrospinal fluid, and in the blood, and we can use that to, we can detect that. So it is increased in MND, and it's quite high in MND because motor neurons are very long, so they can release a lot of neurofilaments. And at the moment, it's now required to include it in most clinical trials. But I want to highlight that it's increased in multiple sclerosis, Alzheimer's, Parkinson's, frontotemporal dementia, traumatic brain injury, and Huntington's disease. So it is not specific just for MND. So I just hinted a little bit in my previous slide on the diagnostic problem. We can have a biomarker that's diagnostic, so it can help us diagnose, tell us a little bit about how far along in the disease someone is, so that we call that prognostic. And pharmacodynamic is how a molecule responds to drug treatment, and we can then use that in the clinic to determine if a drug is effective or not. most studies, however, look at a single molecule as biomarker, and I just gonna show you a very complex graph to highlight that one single molecule, in my opinion, is not enough for motor neuron disease. So here I'm showing you a very complex diagram, but we have the motor neuron, the muscle, and anything that happens in between, a lot of mechanisms that are affected in motor neuron disease. So I don't think one single molecule can encompass what is going on in MND. But along this graph, I'm just going to highlight where we see fats that are important. So almost everywhere along this mechanism chart, we see that fats play an important role. So how do we get to a biomarker? So we need to use first, we usually go to genetic mouse models, and a lot of you may have heard about the genes that are involved in MND that are SOT1, C9, and TDP. So we have mouse models of these gene mutations, and the beauty of mouse models is that we can take tissue and blood before the disease starts, and we can't do that in humans, unfortunately. And then we can track over time how things change. So another really complex data set, but I just want to highlight that we get about 511 different fats per blood sample. So from these mice, this is a little example about how the fats are changed based on genetic makeup of the mice. So if we look, so the darker the red, like it goes in a different direction, the more blue, it goes in a different direction, up or down, relative to each other. So for example, if we look at this, the far right, you see the brown column, and it's different, and I can actually say this is a mouse with a C9 mutation, and it's towards the end stage of the disease. So you see that like at the end stage that we see a different lipid signature, like fat signature. So then we also look at human samples, and what I want to highlight is that we want to have a MND specific unique biomarker, and neurofilament, as I said, in Parkinson's disease, for example, it's also upregulated. So we took some human samples, blood samples, and had a look at their lipid profile, and very interestingly, we saw in the green, we see MND samples, in red is the control samples, and in blue is the Parkinson's disease samples, and we see that they're very, very different. So that tells me that there is a distinct fat profile that's different in MND patients from Parkinson's disease. So I said I don't want to use one single molecule, so I decided I want to use 3 different fats, all in a different part of a mechanism involved, to encompass the heterogeneity of MND. So we're going to use machine learning, and we had about 500,000 of combinations of lipids to be tested, and this depiction is literally what I was like, this is too much. How am I gonna tackle this? So I did hire a really good data scientist that helped me out to take the fat profile from the mouse, setting rules and threshold and all sort of bioinformatic jargon that I won't bore anyone with. So she managed to reduce from almost 500,000 combinations to 18 million that were potential candidates of MND. So this is the graph, all the 18 lipids that could be a candidate as a potential MND unique and specific biomarker. So now, are these fats actually important in the clinic? So that gets me to my fellowship and what I'll be doing over the next 3 years, and hopefully more if time and funding commits. So this is a graph. So what I want to do is like I get a group of people living with MND, asymptomatic gene carriers. So that's family members of familial people with MND, and also controls. And I want to take their blood and then look at those 18 lipid sets and look at the normal range. So from the control, so see what that range is, and look at an abnormal range from people living with MND, and maybe the symptomatic carriers. We don't know yet because there's very limited information out there what their blood fat profile looks like. And so I'm sorry, I have here lipids, but that's fats. So, and then look at what the normal and abnormal ranges are. So that's ultimately the goal. And what do I then hope to achieve? Is that fats can change over time and can tell us something about disease progression. Fats can be used in the clinic to assess if a drug is effective, but also I hope it can help shorten diagnosis in combination probably with other biomarkers like the nerve conductions or something else. But we like to incorporate this as a generic test that can be ordered through the GP if someone suspects MND. So thanks everyone for your attention. And I'd like to thank Shu for taking me in the last year and supporting my my work. I'd like to thank Professor Brad Turner for guiding me the last 5 years up until this point where I took off and got my own funding. And of course, I'd like to thank MND Foundation, MND Australia, for funding this project. And yeah, thank you. So I think we're gonna leave questions to last. So it is my pleasure to introduce Dr. Azin Amin. I've had the pleasure working with her alongside in the Turner Lab, and she is a brilliant scientist, and it's my pleasure to introduce you.
Thank you so much, Sophia, and thank you, Shu, as well, for the introduction. I'll just...
It's perfect.
Great, thank you. Yes, thank you for the introduction. And also, I'd like to thank MND Australia for giving me the opportunity to present my project at this wonderful webinar series, and also for awarding me with this prestigious fellowship, which allowing me to do my research. So basically, we know that in ALS, there is an accumulation and buildup of these protein clumps, which are also called aggregates within cells, especially within motor neurons, which are cells that control movement in the brain and spinal cord. And it's believed that the buildup of these harmful protein clumps contribute to the death of these cells. So we can use a cell's natural process called autophagy to get rid of these harmful protein aggregates. So autophagy is basically the cell's garbage disposal system. It gets rid of anything that's damaged or harmful to keep the cells healthy. And it's a very complex process, but to simplify it, we can break it up into 3 different steps. In the first step, the cells recognize the harmful components, such as those protein aggregates, and they form a bubble around these toxic components. And this bubble is called autophagosomes. Eventually, these autophagosomes fuse with another compartment called lysosomes. And these lysosomes contain all of these powerful enzymes that can degrade all of these waste products. And basically, this happens in autolysosomes. And eventually, because these components get degraded, any generated molecules get recycled back into the cells to be reused. But what happens in ALS cells is that this system is broken and it doesn't work as effectively. So what happens is that these autophagosomes are able to be formed, but they can't proceed to autolysosome formation. And so in ALS affected cells, we get a buildup of these waste products as well as these autophagosomes. So the aim of my research is basically to find a way to try and boost this autophagy process, and so hopefully get rid of those toxic protein aggregates and protect motor neurons from dying. And this is just an electron microscopy image showing these vesicle structures, which are autophagosomes within a degenerated motor neuron from an ALS patient. So the protein that regulates or upregulates this autophagy process, it's called Beclin-1, which you can think of it as a switch that when it's turned on, it increases autophagy activity. And in my research, we focus on this yellow component of this protein, which is its autophagy activating component. And so using this knowledge, we developed different protein fragments called peptides that are from this Beclin-1 protein. And these peptides are designed to mimic the autophagy activity of this protein. And also we use different strategies to try and increase the activity of these peptides. The reason why we use peptides is because we believe that they may be a great therapeutic agent, and as they are highly selective and they have relatively low toxicity. But they do have a major limitation, and that is that they struggle to get into the cells. In fact, we use the red fluorescence to tag our peptides, and we saw that there was inefficient uptake of these peptides into the cells, as you can see with these red dots. So to help our peptides to get into the cells, we use the helper molecule called ApoE. This ApoE can help transport any substances across the cells as well as across the blood-brain barrier, enabling them to get into the brain. So we've previously used this strategy to deliver another drug, which in here is labeled in purple. As you can see, when we injected the peptide into the tail of these mice, they were able to get into the brain. And in fact, they were able to escape the blood vessels, which are as here in green, to access the neurons in the brain. And so we used a similar strategy for our peptide. But another problem is that anything that gets into the cells, the majority of it will then get entrapped and then will get degraded. And so they won't be able to have an activity. So we used another helper molecule called HA2, which can break the endosomes open and help our peptides basically escape into the cell's interior, where they will then be able to have activity. So we call these ApoE and HA2 basically conjugates CPP. And again, using our microscopy and fluorescent markers, we were able to confirm that when we fused our peptides with this CPP, there was an increased uptake of our peptides or these fluorescents, or these red fluorescents into the cells. So our peptides can now get into the cells. Our next question was whether they can actually increase the autophagy activity in cells or not. So to do that, we used a special fluorescent marker, which will allow us to use microscopy to visualize and quantify autophagosomes as yellow dots and autolysosomes as red dots. And when we screened all of the peptides that we've made alongside a few negative control peptides, rapamycin, which is a positive control drug that increases autophagy, and bifidobacterium A1, which is a drug that inhibits or blocks autophagy, we found two of our peptides significantly increase autophagy activity in these cells. So they can increase autophagy activity. Our next question was whether they can degrade those harmful protein aggregates within cells. To measure this, we looked at mutant SOD1, which is a protein that aggregates and causes toxicity in ALS. In the first instance, we looked at the soluble fraction within these cells. The soluble fraction contains all the normal proteins, and our peptides didn't have any effect on these proteins. But when we looked at the insoluble fraction, which contains all of the aggregated and toxic proteins, we saw that our peptides significantly reduced the amount of these mutant SOD1 proteins. And overall, we found one of our peptides called CPP-BCN4 to be the most effective. So this is the one we took forward to our next experiments. So to confirm the results that I just showed you, again, we used microscopy to visualize these protein aggregates. So in the with the arrows, you can see cells that have all of these green dots or these aggregates. In this one, this is the mutant SOD1. And as you can see, after treatment with our peptide, the number of cells containing these protein aggregates was significantly reduced. We also looked at another ALS-associated protein aggregate, and that's the C9orf72. And again, similar to SOD1, we saw that after treatment with our peptides, the number of cells that had all of these protein clumps or protein aggregates was significantly reduced. So this meant that our peptide can increase autophagy to reduce these protein aggregates, and it doesn't matter what the composition of these protein aggregates are, our peptide is effective at degrading those. So our next question was, does that have any actual beneficial effects on cells? So to measure this, we looked at caspase-3, which is a marker of cell death. So as we anticipated, we saw that cells that express the mutant SOD1 had a higher level of caspase-3 compared to cells that were expressing the normal SOD1. This meant that these cells were undergoing cell death, or the cells were dying. But after treatment with our CPP-BCN4 peptide, this was reduced, which meant that this peptide treatment is protecting our cells from undergoing cell death. And just to make sure that we are doing our experiment properly, we used the negative control peptide, and as expected, it did not have any effect. And astarosporine, which is a drug that induces cell death, was used as our positive control drug, and as expected, it increased caspase-3, which meant that their cells were dying. So given all of these promising results, we next wanted to test the effect of these peptides on mice. So we used a mouse model that again allows us to visualize and see autophagosomes as yellow dots and autolysosomes as red dots. And when we injected these mice and we looked at their spinal motor neurons, we saw that after treatment with the peptides, the number of red dots or autolysosomes was increased in these mice. We also looked at the number of lysosomes, as you can see with the blue dots. And again, we saw that after treatment with the peptides, the number of lysosomes in these spinal motor neurons was also increased. And when we quantified the number of autophagosomes that were interacting with lysosomes, again, we saw a significant increase. So this result meant that our peptide can cross the blood-brain barrier to access the motor neurons and upregulate autophagy activity in these cells. To see whether any of this has any therapeutic benefits, we used an ALS mouse model, the SOD1 G93A. First, we wanted to see whether it has any toxic effects, and for that, we looked at IgG levels, and we saw that our peptides was safe. And next, we injected those peptides over 10 weeks, and we found that our peptide is significantly delaying disease onset and is significantly increasing the survival of these ALS mice. And then we also looked at NFL levels, which is a marker of nerve damage. And we saw that in mice that were treated with our peptide, there was a significant reduction in those NFL levels, which meant that our peptide is potentially preventing neurons from dying, and they are protecting those motor neurons. So given all of these promising findings, we were really excited to move this peptide forward, but we still have a lot more experiments to do, which brings me to my fellowship. The first aim of my fellowship is to further improve this peptide. So we know peptides get degraded in a serum very quickly. So my peptide gets degraded within 30, like its half-life is 39 minutes, which is in line with other peptides. So the first aim is to use different strategies to try and increase the stability of this peptide, so we don't need to inject it repeatedly for it to be effective. The second aim of my project is to test the effect of these peptides on motor neurons derived from familial and sporadic ALS patients to see their effect on protein aggregates as well as neurodegeneration and all the other markers. And in the third aim, I will be taking the most potent and effective peptide and looking at their effect on an ALS mouse model to see their effect on disease progression and survival. And lastly, I'd like to thank everyone in the peptide and oligonucleotide therapeutics group, especially Associate Professor Fazal Shabanpour, and everyone in the MND lab, especially Professor Bradley Turner and Dr. Nirma Pereira, and of course MND Australia for giving me this opportunity to do my research. And thank you all for listening.
Thank you so much, Sophia and Azin, for the wonderful presentations today and your clear enthusiasm for wanting to make a difference for people living with MND. Are there any questions for Sophia or Azin? You can type them in the chat, but there is one from Phil, and this might be for you, Sophia, or Azin. Can we measure the amount of neurofilament that is released from the neuron to measure if disease has slowed or not in its progression?
That is exactly why they are currently in clinical trials, so we can measure it longitudinally in the blood of people living with MND, and to see if the disease does slow down, because then you get less release. So at the moment you can measure it, but yeah, the problem is that it's not specific, I guess. And with neurofilament, it means that neurons are already dead. So you kind of get to a point. It's like, it's not, they can't really come back. So it's really a difficult situation here. I don't know if you want to add to that, Shu.
Yeah, no, I think it is currently being used as the preferred biomarker for MND ALS, but as Sophia and Azim probably know, and I'm sure everyone can appreciate that it isn't specific. And so anything that we can do as researchers to find the biomarker that will tell us more about MND progression, to help diagnose MND and to help track responses in clinical trials would be much better. I mean, if we can diagnose MND earlier, I guess it's a bit of a double-edged sword of receiving the diagnosis earlier, but it means that you can get into clinical trials earlier in the piece. So yeah, there's a lot going into biomarker research at the moment, and we hope that you can find a unique biomarker that consists of 3 lipids.
Yeah.
Dalal, yes, there should be a recording that will be shared by MND Australia, which they've already answered it there. So, Sophia, I have a question for you. You've tested your these lipids in MND against Parkinson's disease, and you can see that it's very distinct. Do you think that once you find what might be the normal range in healthy controls, and you test that against, you kind of use that as the litmus test or the bar for MND, do you think that it would be also worthwhile not just testing Parkinson's again, but also, as you mentioned, because fats are involved in a lot of diseases, same with neurofilament, for example, Alzheimer's, Huntington's, frontotemporal dementia.
I think it would be amazing to test all these lipid, like these fats, in other neurological conditions, and even metabolic disorders, to see if they change and how different diseases kind of like pull them apart, and which one stands out the most. Ultimately, that comes down to funding and resources, but that would be probably, yeah, a great tool to have, and to see how unique they actually are to MND, or if it's actually something else underlying that gets highlighted that causes lipids, no, like fats, to be dysregulated. But we're hoping with a set of 3 to like sort of encompass that variability a little bit.
Okay. Azin, there's a question for you. Have you shown or plan to show target engagement of the peptide in the neurons in different mouse models? Do you expect a different effect in TDP-43 versus SOD1 aggregation?
That's a great question. Thank you for your question. So we've tested target engagement in that autophagy mouse model, where we showed that it increases autophagy, but in our ALS mouse model, this is something we are planning to do. Whether we expect a different response, we don't, because so we think if we can upregulate autophagy, it can get rid of all different sorts of protein aggregates, just like we've shown in our cell culture that it can degrade SOD1 and C9orf72 protein aggregates. We would expect the same thing in different ALS mouse models, basically. But that's why we are doing the motor neurons derived from the familial ALS patients, because we want to ensure that our peptide is effective against all different sorts of those mutations, basically.
Right. There's a more general question about ALS, MND, and that is, are there any genetic changes that could possibly cause or predispose to MND and its symptoms? I think either of you can answer this question, given the models that you're using or have used.
So any genetic predispositions that can cause.
Mhm.
So there are some theories out there that is called like a multi-step process, because there are some genetic predispositions that, as some of you know, that you're a C9 carrier, but you don't even get the disease. So there's a lot, like, I would say in the 50s genes that are associated with MND, but they don't have to even like cause the disease. They can contribute to the development of MND, but there could be other factors like generally aging, smoking, environment. There's a lot of associations with other factors that contribute to that genetic predisposition. I don't know if Evan wants to add something.
Yeah, so following up from that, it's a very complex disease, and although there are, genes that people commonly refer to, and Sophia and Azin have done it today in terms of the mouse models that they're using. So you'll see C9orf72, SOD1, or TDP-43 pop up routinely, and these variants, gene mutations, might be or are linked to MND. I think a lot of researchers in the genetic space would argue that it's not just as simple as 1 or 2 genes. I think the general consensus is that there's probably a lot of genes that might have slight variants, that when all those sort of differences come together in an individual, that increases the risk and predisposes to MND. And as Sophia said, with the 6-hit hypothesis, the multi-step hypothesis, it's likely that that initial genetic predisposition, layered on top of other things that you might come across in your lifetime, will then trigger the onset of the disease. And I think that's what a lot of the field is working towards, trying to understand what those triggers are, because we cannot find a good treatment for this disease if we don't know what's causing it. And to find a good treatment, we need to know all of the causes. So it's great that we have people attacking MND from all angles in the lab. to try and solve the puzzle. Steph has a question. She says, are there any downsides to upregulating autophagy? And is there such a thing as too much?
Again, another great question. So before they used to think autophagy as a double-edged sword, where if you upregulate it too much, it can cause a type of cell death known as autophagic cell death. So as research is coming up, it depends on which stage of autophagy you are upregulating. So if you are upregulating the early stages or the autophagism step, I don't know if you remember, basically the yellow dots, it can cause toxicity, and that's what happens in the majority of neurodegenerative diseases, where you get an upregulation of those autophagisms, which contain all of those waste products. But in our research, that's why we are very focused on upregulating the later stages, which is the degradation part or the autolysosome formation part. And we believe that if you are upregulating that process, especially in disease cells where there is a buildup of those autophagisms, you can rescue the cells. And that's why we did also the caspase-3 experiment to see whether it causes any cell death. And we saw that in fact it was protective. So no, we wouldn't expect downsides with it, especially in disease cells.
Thank you. Azin, I have a question for you. I noticed that in one of your slides you presented that you used rapamycin as a positive control. So rapamycin is FDA approved. It's used in other conditions. It has a pretty good half-life of approximately 3 days, and it crosses the blood-brain barrier. So is there a reason for pursuing your approaches, your peptides, which from your graph look like they had similar effects to rapamycin, which is already available?
Great question. With rapamycin, this is where the comparison between peptides, which I work on, and small molecules come. Small molecules are basically drugs, and they have a lot of different targets. Whereas with peptides, we are hoping that it's going to be a specific drug that only upregulates autophagy pathway. So with our peptides, we are hoping that it will be a very selective and specific drug. And also we are hoping that we could find a more effective autophagy upregulator than rapamycin, because rapamycin's effect also depends on the cell types. So in neurons and in motor neurons, there are some evidence that it's not as effective as compared to other cell types. Whereas we are hoping that we can find the peptide or develop a peptide that can upregulate autophagy specifically in neurons. Does that answer your question?
It does, but can I follow up with another one based on what you just said? So if you want it to be very specific for autophagy, does that sort of discount all of the other things that are going on in the cell? That's working under the assumption that autophagy is the only thing to target. If you remember the picture that Sophia presented, where she showed all of the things that go wrong in MND at the level of the neuron, but also in the muscle, would you imagine then that if your peptide is very specific that it would need to be used in combination with something else to, I guess, help mitigate anything else that might be going wrong?
Definitely, yes. I think it could be used in combination. And also with autophagy, yes, I've presented that it degrades protein aggregates, but it can also degrade damaged organelles such as mitochondria, which can, if we can get rid of those, it can reduce oxidative stress, for example, or it can also get rid of damaged ER, endoplasmic reticulum. So it's kind of, I've presented as its effect only on protein aggregates, but it also could mitigate the other pathological problems that are in ALS, basically.
Right. So maybe not the magic bullet, but close.
Yes.
Sophia, I think this is a question for you from Phil. When people with MND are diagnosed, they're told to go and put weight on and to get fat. The question is getting fatter really an advantage? The process of getting fat quickly usually means eating unhealthy food and doing much less exercise. And Phil acknowledges that this might not be the best panel to ask, but he'd be keen to hear your thoughts.
Thank you. That is a very good point, and we all want to be healthy. And I think it is, then we get into the territory of what kind of fats are we eating to keep those fats on. So we've got some people in our lab that are going to look at which fats are going to be important to help with keeping that fat on. So I think it's not as easy as just go get fat, stop exercising, and be a lazy couch potato. I do think you still need to look after yourself and look at what kind of fats we do eat. But we still have a lot of research to do to confirm which fats you should increase your intake of. So that is to be continued. Hopefully in the next few years we can give you a definite answer on that.
Yeah. And Phil, I think this is a really important question because I think a lot of people are aware that, at least in MND, there's no real dietary guideline of what people should eat, how they should eat, and what diet might actually do in terms of changing the course or modifying things for people living with this disease. So, and this is where the benefit of having dietitians involved in MND research is going to change that narrative. And so, yeah, as Sophia said, people are looking at it. A PhD student in the labs at UQ looking at it. So hopefully we can tell you a bit more. Okay, so there's a Facebook question. Mum gained weight from lack of mobility. Her muscle disposition reduced significantly. She was still overweight. What does the weight gain achieve exactly?
This is quite a complex question without knowing too much, I guess, about the whole situation. Weight gain, if it still depends what kind of fats they are, because when we gain weight, we have different sorts of fat. We have like brown fats and white fats, and they store their energy differently. And the way those fats are used are likely to be different in MND, and how they are metabolized could be different. So what does it higher lipids, it could be protective, even though your muscles are still deteriorating, the muscle disposition reduced. It could still mean that the actual neurons innervating the muscle could still be active, but again, I can't give a definite answer on that without looking into the muscle and looking at how much fat there is in the muscle, where this fat is gained, and how it is metabolized, because that is also very variable between each person living with MND.
And I think the general consensus is that it's, in MND, weight loss is associated with worse outcomes. And, the weight loss being associated with disease is not something that's just unique to MND. We see it in other conditions, when people lose weight rapidly, if they're on a chemotherapy, they're usually told to stop so that they can put some of that weight back on. And that weight really is to give that body the energy that it needs to help fuel the cells. Now, I think at the moment, a lot of people are leaning towards that, eat the fats, put on the weight, because having weight is associated with better prognosis. But I think that also, really brings us back to that concept of the fact that we don't know what is the best approach dietary-wise to help people with MND maximize outcomes. And I think you can put on weight. It's difficult to put on weight if you've lost your appetite. But if you then put on weight and you're still losing muscle and strength, you sort of have this issue of, well, now how are you going to get around when you're a bit heavier if you're getting more weak? So I think it's a very delicate balance that I hope research will be able to address in the coming years. A question about protein aggregates. Do we know if protein aggregates are a cause or symptom in MND? Would completely removing them be expected to halt the disease?
Yeah, that's a great question. The process of protein aggregate formation is very complex, and also it's a very it takes a long time for proteins to become aggregated. Basically, it begins by proteins misfolding and then eventually forming into these little small aggregates, which are called oligomers, and eventually maturing into those large protein aggregates. And in fact, we actually don't know which of those protein species, which of those stages are the most toxic stage, and that's something I'm hoping that through with my project that we can find out. So we don't really know if it's a cause or a symptom. In ALS mouse models, we know that by the time you can visually see protein aggregates, they coincide with neurons dying and the muscles wasting away. And also we know from cell culture studies that the presence of these protein aggregates, these cause harm to the cells. And we believe that if we can find a way to get rid of these protein aggregates, it could potentially slow the disease progression. But with the third aim of my fellowship, this is what I'm hoping to tease out: at what stage of protein aggregate formation process can we target with autophagy for it to be basically effective for disease.
Can I ask a follow-up question?
Yeah, of course.
I think maybe it arises from the fact that, you know, in the field of Alzheimer's, a lot of the drugs that have been developed to target the aggregates haven't really led to really strong outcomes. And I want to ask a controversial question, in that, you know, the first question, the question just before was, is it a cause or a symptom? And you referred to this autophagy process as the cell's garbage disposal system. And if you want to take that analogy a little bit further, if you were thinking of a city, big urban city, like what the neuron is, you've got the garbage truck that goes around the city picking up all the rubbish. And it's picking up the rubbish to keep the city clean. And it's almost as if you put the rubbish at the depot, and when that depot gets overloaded, then you get this toxicity. So do you think that perhaps the initial aggregation process might be a way that the cell is trying to protect itself, to corral everything into one location rather than it being spilled out across the whole city, and then it gets to a point where it tips over?
That's exactly right. So that's what new research is finding, that by the time those large protein aggregates have formed, so those, as you said, those large protein aggregates that you can see, those are actually might not be the toxic species. Those are the way for a cell to protect itself by accumulating all of them into basically compartments within cells. And also by the time that they get to that stage, it might be too big anyways for autophagy to degrade them and get rid of them as well. Yeah, so I guess, yeah, that's what we are trying to tease out on, whether autophagy can actually degrade all of these different protein species, and degrading which of those would have a beneficial effects. As for the Alzheimer's, yes, but unfortunately, the majority of Alzheimer's treatments are antibodies, which are designed to kind of neutralize these protein aggregates. And so that's why we think our strategy could be a bit more effective, where we are trying to degrade them rather than neutralizing. Another problem with antibodies are that they can't get into the cells, so they're mostly targeting the extracellular protein aggregates, whereas we, with our strategy, we can get into the cells and into the neurons and hopefully just degrade them.
Okay, thank you so much. I don't think there are any other questions online or in the chat, and we're getting close to 7 or 8 your time in Melbourne. So with that, I'd like to thank both you, Azin and Sophia, for your wonderful presentations tonight. And I hope that everyone who's been listening online has learned a lot and taken something away from the presentations tonight. And hopefully in 2 years, you can give an update of how things are going. Thank you, everyone, for your time. Have a good night.
You.
Thank you.